Okay. I'm sure we'll get a few students flooding in the next few minutes or so. But if you saw my message that I sent out earlier today, I do want to leave some space, dedicate some of this lecture time today to do Unit 1 review if anyone has any questions and topics they want to discuss. And I could tell from kind of the conversations and back and forth in lab yesterday, it's hard to leave space to take in new content. And so today would be lecture on chapter 5 with Unit 2, but then you would likely, leave that space in your memory to go back to Unit 1 stuff. So rather than trying to overload you with that content, which we do need to cover this week, but I do think we can cover it in one day on Thursday, want to leave space for Unit 1 review. So don't have any formal structure for how I would want this to progress, but just in general, if you guys have topics or specific questions that you want to discuss, just speak up and we'll talk about them. Is there anything you guys would like to review? Was I wrong in expecting to use this time for review? We can jump into chapter 5.
I have some questions. Let me just dial my thoughts real quick.
Yeah, that's fair too. I can give you guys a minute to organize that. Or I guess while you're taking time to figure out any questions and things you might want to discuss from Unit 1, I did look into the questions that we left off with last Thursday about Marfan syndrome. We had a few questions left from that activity. And if any of you guys looked into it and want to share what you found, that'd be great too. But questions we had were, how do patients with Marfan syndrome survive for so long? Like, why isn't the death rate at an earlier age? Or what are the rates of Marfan syndrome being inherited versus developed? So developing that mutation throughout their life versus inheriting it on a chromosome from their parents. And then is there a particular demographic or rate of susceptibility for Marfan syndrome? So did anyone look any of that stuff up?
Not personally.
Okay. I briefly looked into it. I didn't do a rigorous search for this info. But there is actually a good amount of info on Marfan syndrome out there. So found those answers from what I believe would be relatively reliable sources quite quickly. So for the question of how do patients with Marfan syndrome survive for so long, the best response I could find is that the severity of the symptoms and which particular symptoms they are exhibiting varies a lot across the individuals, across patients. And so some of those more severe symptoms, like the weakening of the connective tissue around your aortas that could cause it to burst, would either develop over time or are extremely rare. And so that kind of goes into the last question a little bit, or no, that's not how I had these notes. Anyway, severity of symptoms varies and can develop over time. And so until around the 2000s, the life expectancy for someone with Marfan syndrome was around to the mid-40s. But then in the past 20 years or so, the past 2 decades, that life expectancy has gone up to almost match the general life expectancy, typically around the age of 70 or so, just based on further understanding of Marfan syndrome, further awareness, and thus more diagnoses earlier on, and so more treatments earlier on, and thus an increased life expectancy. So not extremely detrimental nowadays. We do have a range of treatments for it as long as it can be caught relatively early on. So yeah, expected to survive for a general life expectancy of around 70 years. For the question of, is it inherited, or how often is it inherited versus how often is it developed? Seems to be that around 75% of patients that exhibit Marfan syndrome did inherit it from their parents, so on the chromosomes that they inherited. And it is a dominant trait, so you just need to inherit one copy of the trait in order to exhibit it. And so that leaves about a quarter of patients who developed it during their lifespan. So some sort of mutation or some sort of outside factor causing a mutation that led to Marfan syndrome, led to those differences in the fibrillin protein. Yes, Ben?
Yeah, I was just wondering, since you say more diagnoses led to a increased life expectancy of people with Marfan syndrome, I'm wondering if that could be because they were less likely to get diagnosed unless they displayed severe symptoms before. So maybe we just found out more people have Marfan syndrome or something.
Yeah, and even understanding the cause of symptoms, right? Because symptoms can, or multiple diseases, multiple disorders can cause similar symptoms. And so without knowing what causes to look for, it can be more difficult to then provide treatment or to offer the kinds of um, procedures that would be required to determine the actual cause and get a full diagnosis. So yeah, all those things would lead to um, less understanding, less treatment, and thus that shorter lifespan previously.
And I think advances in treatment of aortic aneurysms, I think that probably help increase lifespan because that is one of the chief mortality factors in patients with Marfan's and aortic aneurysms.
Yeah, that makes a lot of sense too. Thank you for bringing that up. Yeah, better understanding of how to treat even the specific symptoms without knowing the full cause of what is causing those symptoms would also help with that too. Yeah, cool. Okay, so yeah, again, about 25% of patients develop it in their lifespan, develop that mutation that changes their fibrillin proteins, and 75% inherit it. And then the last question of interest of, is there a particular demographic that is more susceptible to Marfan's? From what I found, no. There is no known correlation. It seems to be even across male and female sexes, no known ethnic orientation or correlation with it. And so in general, it's like a 1 in 5,000 to 1 in 10,000 prevalence rate for the general population. It's 0.01 to 0.02% of the population exhibit Marfan syndrome. So relatively low or relatively high, depending on what you're comparing it to. But so yeah, those were the answers I was able to find to those questions. So thanks for asking those and providing them at the end of last week's class. It's cool to know. So that wraps up that portion. So do you guys have questions for me on Unit 1 topics you would like to review on Unit 1? Let's see, maybe let's try doing it this way. Let's create a poll.
So, sir, how would we be tested on in terms of histology?
Cool. Yeah, so questions on histology could relate to identifying from an image, given an image of a particular tissue and asked to identify what type of tissue that is. Could be asked based on a description of what is present in that tissue, such as types of cells or types of protein fibers. And what was the other that I was thinking to add there? I think that's really it, honestly, recognizing from an image or from a description. So just generally understanding what are the major components of each of the major categories of types of tissues. So like for epithelia, I wouldn't give you a question describing what's in that tissue because they generally are composed of the same stuff. So recognizing that they're avascular, recognizing that they're polar. So more for an epithelial tissue, you'd be asked to identify the type based on how we name them. So based on layers, based on shape. For connective tissue would be like the components. So a more viscous matrix or a highly fibrous matrix, or very little matrix at all, and mainly just minerals like bone, cell types in connective tissue, and then being able to recognize the 3 different types of muscle tissue. So if you were given an image, can you differentiate between them? Or if you were given a description, could you differentiate between them? And then recognizing the general structure of a neuron is all that I would be looking for nervous tissue. Was that clear enough? address that question.
Do we need to know much in terms of the composition of the matrix of various tissues?
Just the general idea of it. So not the full, I'm not going to ask you to list out exactly what's in the matrix of each of them, but generally recognizing which are more liquid, which are more viscous, which are more fibrous. That's all. Okay, so I'm making this quick poll question. So just to get a quick idea here of how confident you guys are feeling with this Unit 1 content, I'm gonna have you rate it from 1 to 5, 1 being unconfident, I feel like I have no idea what I'm doing, and 5 being very confident. And let's just quickly save that and send it out. Okay, so you guys should all get a pop-up for this quick little poll. Again, just rating your confidence in Unit 1 content, 1 being not confident, 5 being very confident. Still waiting on one more response there. Okay, we've got all of them. So I can share these results really quick. So a good number of 3s and 4s, but a couple 1s and 2s and no 5s. So we are still on this lower spectrum, which means there definitely are topics that would benefit you guys to discuss today. So I do want to do that. I just need you guys to guide me. Where should I be addressing our time? Which stuff are you feeling closer to a 1 or a 2 on that's keeping you away from that 5? You're muted, Constance, if you're talking to me.
Yeah, in terms of landmarks for bony structures, how much detail are we required to know?
Okay, so just to clarify, that will be on the lab exam next Monday, not the one coming up tomorrow. And so as far as how much to know, any of them on that list are fair game as far as being asked to identify them, like with the example I set up in lab where there would be a label attached to it asking you to identify them. So you could potentially see any of them. You're much more likely to see bones and bony markings from bones that we did models for, like we spent significant amount of time with. Yeah, I'm not sure what else I could provide to answer there. Like you're not going to be asked to describe them or draw them yourself. It's all going to be real models of bones in front of you, or I guess fake bones, but models in front of you where that structure is labeled and asking you to identify it. Yes, Jim.
Okay. Yeah, hi. I'm a little bit stressful because to be honest, I'm not quite sure how detailed should we know about each like the knowledge points. For example, like APP, you said page 59 and a figure 226. So after reading the whole page, so we need to know all the knowledge on this page or if something from your point. So yeah, I'm kind of a little bit confused about this. How many things we need to know and how details. Yeah, this is my concern. Yeah.
Yeah. So I need to be thoughtful with how I respond to this. Be careful with it. Because technically anything in the textbook as a part of a chapter that we've covered is fair game. My personal philosophy as an instructor makes it so that I'm much more likely to provide questions based on things that we spent time on together. And I look at this as a team, and it's part of my job to assist us all in reaching a level of understanding. So I stray away from questions that we didn't directly discuss in class. So you're much more likely to see things that we did talk about in class. I can't give you a definitive on whether I did or did not include anything from outside of a PowerPoint or outside of a lecture, but it is all technically fair game, but more likely to see what we talked about in class. And so along that same wavelength with my philosophy as an instructor and how I'm teaching, I've tried to assure you guys most of my question types and most of the questions that I'm asking are not very focused on specifics. There are some questions asking specifics, factual type questions, but I care much more about conceptual understanding, relationships. And so tried to talk about those as much as possible in class time. And those types of questions, yeah. So I know it is a lot of content. Technically, all of it is fair. You are much more likely to see questions related to things we directly talked about in class in the PowerPoints. And I care much more about exhibiting a understanding of a concept than I do about memorizing each of the molecules involved and all steps of a chemical reaction. Yeah. So like for cell structures, knowing the general function of each organelle, or generally what each component would provide to something like the cell membrane, right? The cell membrane we went into detail about. So knowing general function that each parts of the cell membrane provide. Like you're not going to be given a snippet of DNA and asked to transcribe and translate that DNA into a polypeptide with specific amino acids. Rather, you're more likely to be asked about generally how the process of transcription and translation works. So transcribing a piece of DNA into an mRNA transcript, that mRNA leaves the nucleus, finds a ribosome, and the ribosome translates it into a protein using tRNA. That's like a general look at the steps rather than getting into the specifics of A bonds with T or U on mRNA, and that correlates with this codon, which is going to produce that specific amino acid. I'm not going to ask that. So hopefully that general idea can be applied.
Yeah. That makes me feel better. Thank you.
So again, there are some questions that are specific to like little facts, but I'm not going to be pulling some random thing out of a list of facts like that. I hope that makes sense.
Yeah, totally. Yeah, thank you.
Okay.
How much details do we need to know about the enzymes involved in transcription and translation?
You don't.
Great. Good to know.
Yeah, that's definitely part of that specifics side. Yeah, you're not going to be asked to name each of the molecules involved, like mRNA polymerase, and which molecule, which protein unzips the DNA versus which protein adds it. Yeah, not going to get into those specifics. I would say the only process we talked about that we went into enough detail together that I would potentially ask specific questions about it would be the electron transport chain and the production of ATP through that. But like I said, in class, I didn't teach about, and I don't expect you to know each of the steps of the TCA cycle, right? There's whatever, like 12 chemical reactions in that cycle. You're not asked to know all those and the different substrates and reactants or products being produced.
So we had a game for the tissues module that had a lot of images of all the different tissue layers. How familiar and confident should we be with seeing tissue samples and being able to identify the layers? Like, is that visual aspect anything that we should prepare for or like pour some extra energy into?
So yeah, in that activity, you probably saw like a variety of examples of each tissue type that may look slightly different than the figures that we used from the text. On the exam, if you're given an image of a tissue, it should look very similar. If not, it would be the exact same as what we used from the textbook.
Oh, okay, cool. Because a lot of them looked different, and like I kind of struggled to like figure out the different representations of those tissue models for a little bit. So I was, that answers my question. Thank you.
Yeah, so you won't be assessed on that. That may be a skill you would like to develop, especially going into a health field. And so we have those slides of different tissue types from different examples, from different samples, from different organs in lab that you are welcome to use lab time for whenever you'd like to look at those to develop that skill. And we can work together on figuring out what tissue type you may be looking at. And most of them are labeled anyways. But for the assessments, I would be providing images very similar to, if not the same as what we used in class.
Okay, thank you.
Are the images primarily going to be diagrammatic or like light micrograph images or electron micrograph images?
Diagrammatic, like animated versions of the image rather than real images. Good question. That's a really
Could we look at desmosomes and hemidesmosomes, just like the functions and structure of them?
the general structure of a desmosome. I won't ask you to differentiate between the structure of a hemidesmosome versus a regular desmosome, or where they're located. Right, we talked about that in class to give you an understanding of why they're named differently for the location on the cell. But just knowing what a desmosome is, the major protein fiber of a desmosome, since that's kind of what gives it that glue structure.
Okay, sounds good.
Okay, was that clear enough? Did that address the question, or was that just as confusing?
That's good for me, yeah.
Okay. So these are all great and important questions to help you guys better understand how you're being assessed. But are there any of these topics you would like to spend time looking at and talking about as well?
I think we delved briefly into like tight junctions and gap junctions. I'm not quite sure how that would pertain in how we would be tested.
Sure thing. Okay. So again, from a conceptual perspective, which I care much more about, recognizing the function that a gap junction provides compared to the function that a tight junction provides. So they're both adhesion type molecules with different types of proteins that provide that adhesion. But so specifically for this question, like a gap junction provides channels, gaps that allow for the diffusion of molecules between neighboring cells. That would be the part that I would hope you know about gap junctions. That they provide those channels for the diffusion of molecules between them. And then a tight junction does not. A tight junction is completely tight and actively prevents diffusion as much as possible by basically being like a stitch holding those two tissues together. Are there any other
Okay.
more specific questions about that that I might be able to answer? Was that okay?
I don't know if it quite pertains to this perfectly, but what are some of the important electrolytes that we should take note of? I mean, we have potassium, and then there's, salt, but what are some ones that we should note?
Yeah, you should know the major cations intracellularly and that are in the cell, and the major cations that are outside of the cell, extracellular cations. Because later in the class, we're going to be talking a lot about the movements of those ions, and as far as the role they play with signal transduction in nerves and muscles, and the role they play in the actual action of our muscles. But for now, generally where they are in highest concentrations. So the major extracellular cations and the major intracellular cations. And I forget where that slide is specifically, but for extracellular, it should be sodium and calcium. And then intracellular, it's potassium. And then we consider phosphate and proteins as a part of those major cations, just because they do play a role in the membrane potential and changing the charge. But that's all, yeah. Not asked to memorize the full list of minerals, electrolytes that we utilize in the body and where they're located. Really just focusing on those two categories for right now.
Perfect. And then, so in the phospholipid bilayer, we have peripheral proteins and integral proteins. What is the difference of those two types of proteins, like in their function?
And the function, there's not too much of a difference because a lot of the time they're mainly going to be, sorry, it's the wrong PowerPoint. I don't even know if you're seeing this PowerPoint. Let me get to the slide. It's just referring to where or how they're located in the membrane. So a peripheral protein is just going to be articulating with some portion of the membrane, but not completely going all the way across it. Right? Peripheral, be integrated with it in some way, could be connected to another protein like the peripheral proteins you see here. And then an integral protein is actually embedded in the membrane. But they do serve very similar functions. I guess integral proteins are more likely to be utilized with transport compared to some sort of interaction, such as cell recognition, whereas a peripheral protein might be more likely to be involved in an interaction like providing energy for a chemical reaction or activating another integral protein like what we see here. Okay. So this peripheral protein might play a role in utilizing energy from ATP or GTP to then activate the integral protein channel, which would open the channel. But most of that is really FYI. What I would expect you to know is based on that naming, that a peripheral protein is peripheral to the membrane, not completely integrated, and an integral protein would be integrated into the membrane, into both leaflets.
Okay, then along with that structure, I saw in the study guide that it said we should note what the glycocalyx is, and I see the word there. What is that?
The glycocalyx is describing the entire kind of layer or border that is formed by the carbohydrates sticking off of glycoproteins. And so all of these carbohydrates, right, create essentially like a layer or barrier or border that is referred to as the glycocalyx. Forget what the word calyx itself actually refers to, but that might be helpful. Calyx in anatomy. I'm getting a lot of things related to the kidneys. Okay, yeah, I don't think the definition of calyx is going to be helpful in remembering that. In botany, it's referring to the sepals of a flower, and in zoology, it's referring to a cup-like cavity or structure. So for our cases, calyx is the, are you guys seeing, sorry, the cell biology PowerPoint? Are you still on tissues?
No, we're seeing this. Yeah, you're
Okay,
good.
cool. Yeah, so the calyx is just the kind of general area where all of the glycoproteins from the glycolipids would be present. And so the glycocalyx of one cell would interact with the glycocalyx of another, essentially to form those bonds that provide cell recognition or help with some local communication between cells.
Would you please explain a little bit about NAD and FADH?
Cool. Yeah, I would love to talk about our electron carriers. Let's see, the slide may be more confusing than helpful. I guess one way to look at the entire process of cellular respiration, which is kind of what we're also addressing here and discussing the mitochondria, because this organelle has become a specialized organ in our cells for completing cellular respiration. And so one way to think about cellular respiration is that we have our three phases. Right? We have glycolysis leading into the citric acid cycle. I guess they're all here. Glycolysis leading into the citric acid cycle and leading into the electron transport chain. These first two phases, glycolysis and the citric acid cycle, when oxygen is present, are just preparing for the last phase because we get so much more ATP from it. With just glycolysis alone, we only get 2 net ATP. We had to put 2 in, we get 4 out, so we only really make 2 new molecules of energy. And without oxygen, those electron carriers that I'm going to talk about more in depth in a second need to be recycled, and that creates lactic acid, and that needs to be converted back to pyruvate, and that ends up using energy. So glycolysis on its own is a net 0 gain. The body itself does not gain any usable energy from glycolysis. And so really, glycolysis is just transferring energy to this electron carrier, NADH, and then produces 2 new products from that glucose. These pyruvates are going to move on to the citric acid cycle, which is going to make more of these electron carriers. We get 3 NADH and 1 FADH2 per pyruvate. So 6 of these and 2 of these from 1 full glucose molecule. And so really by the end of these first 2 stages, we've only made a little tiny bit of ATP that's new to the cell. And most of the energy that was present in that original glucose is now being carried by the high energy electrons that these 2 molecules are carrying. And so NAD and FADH are both vitamins. I forget exactly which ones. One of them is niacin. The other one's riboflavin, I want to say. Those are 2 B vitamins, 2 of our B vitamins that we get from our diet. And their whole role is to carry electrons, carry energy transferred in these high-powered electrons every time we break a bond between carbons. So that glucose molecule that we started with is a 6-carbon molecule. And essentially these first 2 phases are just breaking the bond between carbons in each of these. And then eventually we release all of our carbon from that molecule as carbon dioxide. So we've ended up generating a whole bunch of these electron carriers, NADH and FADH2. Those electron carriers are what power the electron transport chain or start the process of the electron transport chain. So as far as detail of what you should know about NADH and FADH2, I just want you to know that they are electron carriers. They are essentially intermediary energy carriers for cellular respiration. They carry the energy in the form of high-powered electrons. Those electrons get donated to our electron transport chain, which causes the chain reaction that leads to the generation of ATP. So an electron gets donated to these cytochrome proteins. Those cytochrome proteins will pass the electrons to the next protein in the chain. And that is only possible when oxygen is present. Right? We talked about oxygen being extremely electronegative, so it has a strong pull on electrons and will basically drive them to move from cytochrome protein to cytochrome protein until they eventually end up on that oxygen and create water. Every time an electron moves from one protein to the next in this chain, protons get pumped out of the matrix, so out of the very center of the mitochondria, and into this intermembrane space. And so we end up with a whole bunch of protons, a high concentration in this space. They want to diffuse back to the matrix where they started, and they can only get there through this channel within the protein ATP synthase. And so they flow through this protein like water moving through a water turbine, a hydro pump, and that causes a bunch of kinetic energy, which allows this protein to spin and drive enough force to shove a phosphate onto ADP, making new ATP. And so if oxygen is present and we can go through all 3 of these phases, get all the way to the electron transport chain from one glucose molecule, we can make around 36 ATP. if we don't have oxygen, if this process doesn't take place, we don't use our electron carriers. We only make 2 net ATP in that cell. And the body overall doesn't make any new energy because we have to recycle these electron carriers and recycle the lactic acid that is formed because of it. So I included a lot of detail there to give the whole story, but maybe trying to go back and specifically answer, you asked what are, or can you have more specifics on NAD and FADH2? They're just electron carriers, intermediate energy for cellular respiration, and they power the electron transport chain.
Thank you.
You're welcome.
Do you have a slide nearby of the composition of ATP or ADP?
Yeah,
I get the triphosphates, but the adenosine I would like to see.
absolutely. So we have that adenine, which is a nucleotide. Right? We have a ribose sugar, and then our 3 phosphate groups coming off that ribose sugar. So it looks somewhat similar to a nucleic acid. Right? We have a sugar with our nitrogenous base. So we
Do you have
have,
it pulled up?
yeah, is it not showing for you
Oh,
guys?
no, we can't see it.
Okay. Let me just share this whole screen so I don't have to continuously check on that. Sorry about that. So let me go back to where I started. This is ATP. Very, very similar structure to a nucleotide or a single nucleic acid. Right? We have a ribose sugar acting as our base, an adenine, which is a nucleotide coming off of it, and then 3 phosphate groups. So a nucleic acid would only have a single phosphate group being utilized to make the bond with the next one in this chain. Phosphate groups bond with the ribose sugar of the previous nucleotide, etc. And so ATP is kind of like a modified nucleic acid. Instead of only 1 phosphate group, we have 2 or 3, ADP for 2, ATP for 3.
Thanks.
Welcome.
And then with a protein, they have their amino group and the carboxylic group. Can I just see those real quick?
Absolutely. So yes. So every amino acid has the same general structure. There's a central carbon, and each of the available bonds on that carbon is being given to a different side group. So one bond is going to have oxygen. One bond is that carboxyl group. The other bond is the amino group or amine group, which gives this molecule its name, the amino acid. And then the last bond is going to be what changes between the 20 amino acids that we utilize in our bodies. So that last bond is referred to as the R group because it varies between amino acids. And I don't have a table in this slide deck showing each of those. So I guess you'll have to take my word for it on that, or I could go find a table if you want to see what those different side chains are. Because we have amino acids that we categorize based on the chemical properties of those side chains. Some of them are polar, or create strongly negative polar molecules. Some create strongly positive polar molecules. Some are neutral. Some have both or can interact with other polar molecules on both edges. But in general, the structure of an amino acid that you'd be asked to recognize is this. So look for a centralized carbon with 4 branches coming off of it. Look for a nitrogen on one side and a carbon with double bonded to an oxygen on the other. I guess whatever you use to recognize a carboxyl group is fine, but I typically look for the nitrogen and the 4 branches to know that I'm looking at an amino acid.
Do we need to know the structure of the peptide bonds?
What do you mean by the structure of the bond?
The molecular structure of the peptide bonds?
just as far as like what is bonded to what, like no. So I think my answer is no. Generally, you should recognize hydrolysis versus dehydration synthesis, what those mean and what they're referring to, because this is how we synthesize and depolymerize these molecules, either by adding or removing water. So dehydration synthesis is removing a hydrogen from one molecule and a hydroxyl group from the other. So creating one full new water and creating two available bonds now. We have one bond on this carbon available, one bond on this nitrogen available, and they can now bond with each other. So dehydration synthesis removes the water molecule to allow two molecules to bond to become larger or create one larger molecule. And then hydrolysis is the opposite. Hydrolysis would be using a water molecule, adding it to this, which would separate that bond, break those bonds, and then replace them with a hydroxyl group and a hydrogen. So knowing these two terms, yes. Knowing any further detail about this bond besides its name, no.
Do we need to know the molecular structure of glucose and the other monosaccharides?
No, as far as differentiating between them or being able to draw it, no. Knowing the ratios that we talked about of 1 to 2 to 1 for carbon, hydrogen, oxygen, and a carbohydrate is going to be important, because that's what makes carbohydrates polar compared to a lipid, which is mainly non-polar because of its long chain of just carbons and hydrogens. And beyond that, I don't think we talked about anything else. Most monosaccharides are 6 carbons, but I don't think we even addressed that. So don't need to worry about that. Mainly this ratio. They're typically in a geometric shape. Should be something to look for when trying to identify it, but this is also technically a monosaccharide. Glycerol in fatty acids is technically a monosaccharide.
One of the questions from the study guide says, using drawings, explain why polar covalent molecules are found in water. Is that are the only bonds like in water going to be polar covalent, or I don't know, I guess I'm just confused by the question.
Sure, fair. So yes, both of the bonds present in a water molecule are polar covalent bonds. This is still the chemistry slide. Great. Okay. So we just have 2 bonds present. We have oxygen bonded to hydrogen and oxygen bonded to hydrogen. In both of those scenarios, oxygen is much more electronegative than the hydrogen that it's sharing electrons with, and thus is going to have a stronger pull on those electrons, which will leave both hydrogens with a slight positive charge and the oxygen with a slight negative charge. So both of these bonds are polar. As far as drawing that, we do it. Is that structure shown here? Kind of. Yeah, we didn't ever go over an example like this, but when drawing polarity in a molecule, so if this is our oxygen, these were our hydrogens. Polarity is typically signified with this arrow that has kind of a cross on it, like that, to show the direction that the electrons are being pulled. And then we use this lowercase, I guess I don't remember if it's a gamma or a beta, to signify a slight charge in a polar molecule. These would both become slightly positive. Because their electrons are pulled away, this becomes slightly negative because the electrons are pulled towards it. You signify the movement of those electrons or the pull of those electrons with this cross arrow. We never did that in class. We haven't talked about this. This is the first time you're seeing it as far as this content is concerned. So you would not be asked to do this. But being able to describe why a water molecule is polar and what interactions it is able to have with other molecules because of that, you should know.
Perfect.
So there's that little lowercase gamma.
And another question. If anyone else has a question, feel free to go. But I was just gonna ask about the primary, secondary, tertiary, and quaternary structures of proteins. If we could just review them as, a whole.
Yeah, so the level of complexity goes up with the number associated. Right? So primary being the most simple structure. And so this is how these polypeptides, how these proteins are leaving the ribosome. Right? This structure, in its primary structure, would be referred to as a polypeptide. Protein is typically referring to the functional form of these polypeptides, which would be tertiary or quaternary structure. So the primary structure is referring to just the chain of amino acids that leaves the ribosome. So the ribosome is adding one amino acid at a time, creating that peptide bond between each amino acid, and then it leaves as just a free chain, essentially. There could be some of the associations starting to form to create the secondary structure early on, but most of these are going to fully form or become evidence once the amino acid chain has been released from the ribosome. So the secondary structure comes from, it says bonds, but you should really think of these as interactions. These are hydrogen bonds, right? The associations between polar portions of the amino acids or strongly negatively or positively charged portions of certain amino acids, creating that magnetic attraction, which is going to lead to an alpha helix or a beta pleated sheet, like what you see here. So the secondary structure are from those magnetic attractions, the polarity of the amino acids.
We can only see the screen of the calyx.
See
Here,
the
we
screen
can
of the
see your
calyx?
Google. We're looking at your Google right now.
That's not what I wanted you to see. Okay, screen 2 versus screen 1. Sorry, you would think I have more awareness and practice with this. Are you seeing the PowerPoint now?
Now we are, yes.
Cool. So there's that primary structure, again, the chain of amino acids with their peptide bonds. The polarity of certain amino acids will start to form those magnetic attractions, these hydrogen bonds, creating the alpha helix and beta pleated sheet. But this protein is still not functional and cannot perform its role, bond with whatever reactants it may be responsible for, et cetera. So then protein maturation is referring to fully folding it into its tertiary structure. So most proteins are functional on their own as a single polypeptide that have been folded into their tertiary structure. There would be some stronger bonds formed. Typically is going to require another protein to help with this folding, to hold them into that shape. And they would typically have the shape that they would need to bond with a particular reactant and have the right chemical composition in their active site that would bond with that. So tertiary should be an active protein. A lot of our proteins are also existing in this quaternary structure, which has multiple polypeptide chains interacting together. So most of our fibrous proteins that we've been talking about are multiple polypeptide chains that have interactions with each other, forming together one functional protein from those multiple chains. So yeah, does that answer your question, clear up any confusion you had there, or are there more specifics I can get into?
No, that helps.
Okay, cool. And so yeah, when a protein is denatured, so if it's exposed to a temperature range that it cannot withstand or a particular pH that it cannot withstand, typically a protein will denature, which is referring to kind of reversing this folding process. It'll start to lose its tertiary or quaternary structure, and interrupt these hydrogen bonds all the way back to a primary linear chain of amino acids, which is really important for digestion, but bad for the function of your proteins in your cells.
Are there, do nucleosomes relate to this at all, or are those more just in the cell?
So nucleosomes are referring to proteins being used to protect nucleic acids. I think that's in the cell biology PowerPoint. Let me make sure you're seeing this. Boom. Let's go back to the nucleus. Okay, wrong direction. Nucleus is at the end. Here we go. Right. So a histone protein would be a single polypeptide, so a tertiary structure of a protein. Multiple histone proteins interact together. I don't know if this core with like 8 histones functioning together to make one nucleosome, if that would be referred to as one protein, those histones together. I don't think it is. I think you would identify each individual histone. But the nucleosome structure itself is these 8 histone proteins with that double-stranded DNA wrapped around it. And then those will wrap even further and make interactions with other nucleosomes, creating these supercoiled regions in our chromosomes, which is performing 2 functions. It's protecting the DNA, the nucleotide sequence that is within this DNA, so it's not as exposed to external factors that would change it, cause a mutation. And it also plays a role in our genetic expression. So when our genes are not being actively expressed or will not be expressed often, they will stay wrapped up into these nucleosomes. They'll stay wrapped around those histone proteins. And then we can unravel some portions of that to express it, or we do have DNA that stays more loosely packed with these histones or not packed at all if they're going to be expressed very commonly and need to constantly be accessed. But the nucleosome itself is 8 histone proteins with that double-stranded DNA molecule wrapped around it.
So nucleosomes are generally just going to be the storage form of DNA?
Yeah, the like protected, stored form of DNA, sure. And it's what's making up the structure of a chromosome.
Are centromeres and kinetochores molecularly different?
That's a good question that I don't think I know the answer to. Did you say centromere or centrosome?
Centromere.
Oh, so yes, they're just referring to 2 different things. So one centromere is referring to both kinetochores connected for those sister chromatids. So we have 2 kinetochores, 1 kinetochore in each chromatid, and together they create a centromere. And off the top of my head, I don't remember what type of protein makes kinetochores. So I thought you asked if centrosomes and kinetochores are molecularly different and would not have been able to answer that. I assume so, but.
I think I exhausted all my questions. Thank you.
Good, that's the goal. Anything else you guys can think of?
Not for me.
Okay. I'm gonna try this really quick. See how you guys are feeling now. Did we improve any of your confidence? Looking like we got at least some shifting closer to that 5. So I'm glad it was somewhat helpful. And again, anyone who's on that 3 or below, if you think of any kind of question that would help you get closer to that 5, please let me know. If you're unable to or uncomfortable with thinking of or sharing those questions in class today, please keep in mind I do have office hours tomorrow before our exam, 9:30 to 11:30. And outside of office hours, anytime you guys have questions or would like assistance, please reach out to me. And if you want to schedule something like a Zoom call or an in-person meeting at a different time, we can try to do that too. Something I did want to talk about today is something I meant to address in lab, which is basically the structure or policies for how you guys will enter the lab on an exam day. So before you come in, please have your phones, any kind of technology turned off or on mute and put into your backpack or bag, whatever you carry your stuff in. As you enter the room, you can just leave your bag in the front of the classroom, kind of like behind the desk where I stand or under the whiteboard, if that's more clear. And so you should only have with you something to write with when you go to your seat. Your seat will, yeah, your seat will likely be somewhere, or your name tag will be set out somewhere probably different than where you typically sit in lab. So please just find your name tag. That'll be especially important for the practical exam, the lab version of the exam, since we'll have stations set up in different areas. But for the lecture portion, I'm just going to try to spread you guys out and maybe separate you from people that you talk to most for that. So find your name tag. So please have technology put away, including smartwatches. And please put that in your bag as well. So have technology put away in your bag before you come in. You'll leave your bag at the front of the classroom under the whiteboard before you go find your spot. Just bring something to write with you, find your name tag, and that's where you'll be seated. And I will pass out the exams basically as soon as people are seated. And I will plan to and intend to have the door open before 2 p.m. to ensure that the exam can start at 2 and you can have your full hour and 50 minutes of class time to work on it. I don't expect this exam will take that long, but you do have that time allotted for you. And yeah, I think that was it. Is that clear for everybody? Are there any points of clarification that I can provide for that? Okay. So yeah, I'm just going to plan to continue to leave these 20 minutes or this last 20 minutes that we have available for review questions if you have them. If it seems like no one's asking questions and people would rather leave, that's fine. But we do still have this time dedicated for me to help you out.
I didn't hear it completely properly. So you said we leave our backpacks and jackets up by you. Was that what you said?
Yes.
Okay.
Thanks.
So for the tissues part of our exam, do you or anyone else in the class have a way of remembering at least the names of all of them? I found once I was going through the lecture myself, I was like, oh, I know what this looks like, but if I were given this by itself, could I identify it? And I just had this issue where there were simply so many terms that I kind of know and I kind of understand the general connection to it, but the specific term was not coming to my head. So any way to just cement it besides just your regular repetition or flashcards, maybe a video or a mnemonic.
Yeah, so I personally don't have any mnemonic or memorization technique besides just the connections and patterns that my brain makes. I'm sure there are YouTube videos that have a song or a mnemonic like that to find publicly available out there. But yeah, if anyone in the class has found or has created memorization techniques for tissue types that they find helpful, please share.
Well, I don't have a mnemonic or anything, but I don't know if you've already watched it or not, but I found the crash course video that's about tissues to be useful. So
yeah, I don't think he gives any mnemonics, but he does a similar thing to what I try to do of the word association and meanings of roots and meanings of full words where they stem from to try to help with that. And so, yeah, hopefully with epithelia, at least, like I know there's a lot of different ones that we talked about, but because they are classified with such specific criteria, where we have our shape and layers being used to name them, and they're named by naming the layers first and then the shape. Hopefully that's relatively clear. Like the name of the shape is describing what the shape actually looks like. Squamous refers to scale-like or squished. So the squamous epithelium will look really, really flat. Cuboidal is typically almost a full cube in shape, and then columnar are those much longer columns.
Yeah.
And then the second
Yeah,
part of the name. Go
the
ahead.
photos are helpful for sure. And then like, so I use like the online textbook, and it has some information that I don't remember us talking about from lecture, but I think has like thrown me a little bit. So like under simple squame, we talk about how those cells are part of absorption and secretion. But then if you use the book a little bit more heavily, they talk about goblet cells and how they're like single-celled glands that secrete, and they have different names when you're interacting with different portions of the body systems. So
Okay.
I think adding in like that other resource kind of like threw me off a little bit, because like our slides are super straightforward, and like the little icons where like the body locations are where you would see them most often is super helpful.
Oh, yeah.
Um, yeah, it's just like such a quick, instead of reading like all of this, or like sometimes when you're learning and like trying to condense your information, the very specific terms like pleural and pericardial can be distracting.
Yeah.
So I found the icons to be super helpful, but there's just like some detail that I've read in the book, I think that's kind of like throwing in a wrench for me, because
Yeah.
it seems more detailed than maybe what we need.
Yeah, so things like that, like terms for particular cavities where we're not necessarily going to spend a lot of time with in this class or spend a lot of time referring to those things often. I just show reference to it, like the peritoneal cavity in the skin lecture I was working on, whatever, talking about of course, the name is escaping me right now. But yeah, you're not going to be assessed on that as far as a location, a peritoneal cavity. And for most of these, 99% of these, you're not going to be assessed on where they are unless we like really heavily emphasize something like stratified squamous epithelium being a major part of the epithelium of our skin. Okay? We're about to go into a full chapter on the skin, and so made sure that I touched on that as a specific example. Beyond that, cuboidal epithelium being parts of glands, as we're going to go into some more glands. It's really all to focus on there. So yeah, I guess if for names of cavities and stuff, you don't need to know that. So if that's overloading your mental, overloading your memory, try to push that out because you won't need it for this assessment.
Okay, that sounds good. Sounds better.
All right. I'll give one more minute to see if anyone else has questions, and then I'll close out so we're not holding everyone here. So thank you guys for coming today, for participating in this. I hope it was helpful for everyone. Again, if you have further questions you'd like to discuss, please reach out or come to office hours tomorrow morning, if you're able to. And other than that, I will see you for our exam at 2 p.m. tomorrow in our typical lab room.
Thank you.
Thank you.
Thank you.